Well, on the topic of future and what exciting things may come, you talked about artificial organs and there's um, yeah, I think interesting developments there. There's also, I was at the Society for Lab Automation and Screening SLAS event a couple of months ago. A lot of talk about autonomous labs and robotics. I took a tour recently of Ginkgo Bioworks' autonomous lab, and they were speaking with Jason Kelly. He was talking about a recent partnership they had with OpenAI, where they gave GPT-5 access to this lab and reduced the cost of cell-free protein synthesis by some, they said, a significant margin, as maybe a small test case of what can happen when you marry kind of AI and intelligence with, physical automation and autonomy. I'm curious, you're, again, in my understanding, as not a biologist, you're kind of more upstream of that and understanding, affinity and binding with proteins. At the same time, though, you did say, well, the way in which these models improve is through access to, more data as through, high throughput screening and so forth. I imagine something that these labs could be, well designed to do, and maybe there's some sort of feedback loop there. So I'm curious, how you think about autonomy and automation and where you see a marriage between that and what you're building.
Yeah, no, I think they are very, very closely related. And, as I was saying, I think, there is a bit of a dream, eventually of having, the field calls zero-shot drugs, where, you can put in, your target product profile on these computational tools, and then you basically get out your, preclinical candidate, out of the model.
Yeah.
We're nowhere close to that. And so in practice, what that means is that you're still going to need to have, some of those kind of iterations. And those iterations inevitably pass from, having to test experimentally, do some experiments in the lab. And so, that iteration loop, sure, we'll be able to reduce the number of iterations, we'll reduce the size of each of those iterations, but then, how fast you can iterate on those loops and, feedback, the data into these models will be critical. And so I think, kind of all the advancements and, kind of autonomous labs are incredibly exciting from kind of the field. I think, I am not an experimental biologist, and but I when I was here at MIT, as part of my PhD, I did take some experimental biology classes, and coming from a world of software engineering and machine learning, where we often complain about reproducibility and noise within experiments. But kind of when you look at kind of biology and experimental results that we get in biology, we're talking about a completely different level of lack of reproducibility, of noise. And so I think there is so much, I think, good in the field that can come from really kind of not getting to the point that kind of these autonomous labs can kind of do, as well as some of the amazing kind of incredibly patient human biologists, experimental biologists, and experimental chemists, but hopefully even better and a larger throughput and so on. So super excited about kind of all of that field.
So we've talked about artificial organs, automated labs. I was recently learning about, to the point of clinical trials, implicit approval regulatory environments, where I think in Australia and in China, I think a clinical trial is approved within 60 days, unless, proven otherwise, which seems to increase the throughput and without a significant impact on safety, which is great. I'm curious, as you look to the future, what technologies, regulations, policies are you excited about that you think are really going to move the needle in this space?
Yeah, I think one of the things that we are working on, and I think with the help of kind of these autonomous labs, we're going to really be able to push through is kind of this idea of being able to go from a therapeutic hypothesis. So as we were saying, a scientist having a hypothesis, okay, this target might be related to kind of this particular disease, to having a clinical, preclinical ready, lead kind of candidate, and kind of significantly reduce and accelerate that time thanks to kind of these models that we're building, the autonomous labs, and these few iteration loops that are going to happen. Now, kind of, when that happens, I think, there's going to be an incredible kind of wealth of, a lot of different ideas that, people have had, maybe in academia, maybe around, diseases that are kind of less funded than others that so far have not been able to be kind of tested, and, they're going to be able to kind of go through and, be tested, for example, in animals and so on. But then I think, one of the things that we might see is that, we are going to hit kind of a big bottleneck that comes into typically, phase 2 clinical trials. That's really, phase 1 is something that we can reduce. It's really about kind of, toxicity. That's something we can, largely, I think, kind of going to be able to model. But phase 2 is really, is also around, was that the right target? And so it's really kind of, this somewhat testing at the deeper level that therapeutic hypothesis. And so that's, I think, where there is a lot of also kind of, exciting research that luckily kind of, many other folks in the field are working on around things like, the virtual cells, or in general, even, ways, whether it's via virtual organs or via other things to have, better models of, kind of, human kind of efficacy and, kind of disease that will allow us to, basically be able to, close this loop, without, needing to invest, tens to hundreds of millions of dollars into a phase 2 trial before, learning that an hypothesis wasn't correct.
Well, I'm going to ask you to make a very easy prediction. Should be very simple for you. We're in 2026 right now. By when will we have a Boltzgen-generated molecule that is delivered to a patient to treat an illness?
That's a great question. Well, if we were to, based on, usage, if we were to, kind of take an estimate, it would definitely be, 5-10 years from now, because that's, kind of the time that it takes. I don't know that we're gonna, kind of the molecules that are being created these days with Boltzgen, they are, passing through in later stages of clinical development, will benefit from some of those acceleration of, preclinical clinical trials that we have talked about. So we might have to, wait that long. But yeah, hopefully, I mean, if AGI comes in 2 years, everything else will be easier.
There we go.
Jokes aside, I think, hopefully can be accelerated, but, in general, I'm definitely very excited about, yes, kind of what's coming in, 1 or 2 years, but I think, over the next, 4 or 5 years, we can really kind of, get to a point where we can really kind of reduce a lot of these kind of bottlenecks that we talked about.